Hello, I'm Axel Nothoff. To study the coordination of different lipid synthesis pathways during membrane biogenesis, it's useful to have an experimental system where membrane biogenesis occurs rapidly and inducibly. We found that phagocytosis of latex beads is particularly useful for such studies because cells very quickly synthesize new membranes to replenish those that were lost as wrapping material early during the engulfment process. In this video, my colleague Xiaochong Zhang will demonstrate procedures we use to study phagocytosis-induced changes in gene expression using a luciferase-based reporter gene assay approach. Where's Xiaochong?
Hi, I'm Xiaochong Zhang from the Laboratory of Axel Nothoff in the Department of Molecular and Cellular Biology at Harvard University. Today, I will show you how to use the reporter gene assay to analyze the regulation of genes involved in membrane biogenesis after phagocytosis. So let's get started. For this procedure, we use cultures of human embryonic kidney 293 cells. These cells are grown in 10 cm culture dishes in medium A, which consists of tubercles, modified Eagles medium or DMEM, supplemented with 10% fetal bovine serum or FBS and a cocktail of antibiotics. Using a standard trypsinization technique, the cells are detached from the bottom of the dish, counted, and then plated on polylysin-coated 96-well plates, where they are grown for 24 hours until beginning transfections. For our transfections, we routinely perform all conditions in triplicate and transfect a total of 50 to 100 nanograms of plasmid DNA per well. Plasmid mixes should include at least a reporter construct expressing firefly luciferase and a control plasmid expressing renilla luciferase from a constitutive promoter. To begin, prepare a mixture of serum-free and antibiotic-free DMEM with transit 293 transfection reagent. Use 10 microliters of DMEM for each well to be transfected and add 3 microliters of transit 293 reagent per microgram of plasmid DNA. For example, if 20 wells of each to be transfected with 50 nanogram DNA, add 3 microliters of transit 293 reagent to 200 microliters of DMEM. The solution is mixed by repeatedly pipetting up and down, and then left undisturbed at room temperature for at least 5 minutes. Next, the DNA is pipetted into separate microcentrifuge tubes. The concentrations of our plasmid stock solutions are usually between 10 and 100 nanogram per microliter. Once the DMEM transit mixture has incubated for 5 minutes, it is transferred to the DNA tube and the solution is again mixed by pipetting. Then let the samples stand at room temperature for 20 minutes. During the 20-minute incubation, remove the media from the 96-well culture plate and replace it with 90 microliters of fresh medium A. Once the incubation is complete, add 10 microliters of the DMEM DNA transit 293 mixture to each well. The mixture should be pipetted directly into the existing liquid. Don't let the pipette tip touch the wall of the tube. It is also important to include 3 wells of cells that will be left untransfected. The average luciferase activity in these wells will later be subtracted as background. place the 96 well plate into the 37 degrees C incubator and grow the cells for 24 hours. Once the 24-hour incubation is complete, we proceed with inducing membrane biosynthesis via phagocytosis of latex beads. To begin, prepare a suspension containing cell culture medium and latex beads. To do this, prepare medium B, which is a 1:1 mixture of DMEM and Hem's F12 medium, plus antibiotics and 10% FBS. Then add 0.75 micron latex beads at concentrations of up to 1 mg per milliliter. Of course, every experiment should include controls where no beads are added. Next, remove the media from the 96 well culture plate and replace each well with 100 microliter of the bead suspension. Once the suspension is added, spin the plate for 2 minutes at 1000 G. This helps concentrate the beads at the bottom of the wells and facilitates phagocytosis. Next, culture the cells at 37 degrees C for 6 to 16 hours, depending on the conditions and the promoter used to drive luciferase. Increased promoter gene expression can be detected after 1 to 3 hours. For certain experiments, it might be necessary to uncouple phagocytosis from lipid synthesis. For example, to study the effects of certain drugs on lipid synthesis without affecting particle engulfment. In such cases, incubate the cells with the beads for 30 to 60 minutes to allow cells to phagocytose the beads. After this incubation, the cells are washed 2 times with PBS to remove unincorporated beads. Then fresh media is added and then cells are incubated for 6 to 16 hours for reporter gene expression. Now we are ready to proceed with the luciferase assays. To begin the luciferase assays, prepare a lysis buffer containing 20 millimolar trace HCl, pH 7.8, 10% volume-to-volume glycerol, and 0.5% volume-to-volume Triton X100 prior to use. Adequate amount needed for the experiment. Then add 1 microliter per milliliter of 1 molar DTT. Do not reuse the leftover DTT solution. Then add a protease inhibitor cocktail to a final concentration of 0.5 to 1 times. When using the dual glow kit for the first time, prepare the Firefly luciferase reagent by transferring the entire contents of 1 bottle of dual glow luciferase buffer to 1 bottle of dual glow luciferase substrate. Divide the unused luciferase reagent into 10 ml aliquots and store at -20 degrees C. Once the luciferase reagent is ready, remove the 96 well plate from the tissue culture incubator and place it on ice. Next, remove the media by aspiration. Then add 40 microliter per well of lysis buffer and leave the plate on ice for 30 minutes. Next, aliquot 15 microliter of luciferase reagent per well in a white opaque 96 well plate we use Perkin-Elmer's OptiPlate 96. Then add 15 microliter of cell lysate and incubate the plate at room temperature for 10 minutes. After 10 minutes, read the luminescence in a microplate reader. Next, perform the granular luciferase assay in order to obtain data for the control plasmid. To do this, prepare immediately before use appropriate amount of granular luciferase substrate solution by adding 1 part dual glow stop and glow reagent to 100 parts dual glow stop and glow buffer. Next, add 15 microliter of substrate solution to each well. Incubate the plate at room temperature for 10 minutes and then measure the luminescence again. Please note that we modified the manufacturer's recommended protocol for these dual glow luciferase assays in order to reduce the amount of reagents required per sample. Now the plate can be analyzed again using a plate reader. Once the data have been collected with the microplate reader, they can be transferred to a spreadsheet program such as Microsoft Excel for further analysis. First, calculate the average from the 3 untransfected wells and subtract that value from each of the data points. Next, divide for each transfected well the value for firefly luciferase by that for granular luciferase, and then calculate. Then, for each set of triplicate samples, calculate average, standard deviation, and coefficient of variation. In our example, we used different concentrations of beads, which have been plotted on the x-axis against the ratio of firefly over granular luciferase activity. As you can see, there is a dose-dependent increase in this ratio. We have just shown you how to use a luciferase-based reporter gene assay to measure expression from the LDL receptor promoter in response to phagocytosis of latex beads. When doing this procedure, it's important to remember to be sure that the cells are only 80 to 90% confluent at the time of transfection. So that's it. Thanks for watching and good luck with your experiments.